I will be talking about engineering the follicle environment, and the major focus in my lab is designing functional and tunable hydrogels for reproductive tissue engineering. So there is a lot of follicles on this slide. So I will be talking about ovarian follicles and then maturation of follicles, and also some of the work that we're doing for follicle implantation, in ovarian tissue implantation. The major motivation for the work in my lab is fertility preservation for either cancer patients or women who face premature ovarian failure. As you probably know, at cancer diagnosis, a lot of anti-cancer treatment cause sterility, or they affect the follicular pool. Women of age, so adult women, can undergo ovarian stimulation, and then they can have their eggs or fertilized oocytes cryopreserved for future pregnancy. But there are a large population of women who cannot benefit from this treatment. So if you think about women who have breast cancer, they cannot really undergo stimulation. Girls and women that have urgent cancer that cannot really wait 2 months to start their anti-cancer treatment. And then the large population is prepubertal girls. So if you think about girls who have leukemia when they're 2, 3, 5, before puberty, usually the treatments that they're given make them sterile. So by the time they have to undergo puberty, they don't have any ovaries to push them through puberty. So this is one problem with this population, and obviously they're also sterile, so they cannot have children. So this is the clinically available option, which doesn't really work for prepubertal girls and other populations. So what is there to offer? So one option is to try and grow those ovarian follicles in vitro. So ideally, before the anti-cancer treatment, the ovary will be removed, and then we can isolate some follicles from the tissue and then try to grow it in vitro, bring it to the oocyte stage, and then cryopreserve it. Alternatively, we can cryopreserve the whole tissue, and then when this woman or a girl is ready to have children, we can transplant the whole pieces of tissue, and then she can have children, and also the ovarian endocrine function is going to be restored as well. One caveat here, although if you think about leukemia patients, leukemia is a blood-borne cancer, so it's everywhere. So you cannot really take the whole piece of tissue and put it as is, because you will be putting this person at risk of reintroducing cancer cells. So I will show you some ideas that we have to try and overcome these limitations. So I know a lot of people here work in reproductive biology. This is just kind of follicle 101. Beautiful structure of ovarian follicle. Follicle is a unit. There is an oocyte in the middle surrounded by granulosa cells, antrum, and then some theca cells on the outside. Ovarian follicles can be found in different developmental stages. The majority of the follicles are primordial follicles, and it's really difficult to culture them. There is no real facts in the literature showing that somebody can really culture primordial follicles and bring it successfully and efficiently all the way to live birth. So the majority of work done on primordial follicles is implantation and growth in vivo. There is successful reports of culturing follicles in vitro, where primary or secondary follicles can be cultured in vitro and then brought all the way to ovulation and even live birth. So how can we culture follicles in vitro? So obviously we can isolate follicles and then just put them in the dish and let them grow. And then mice follicles do grow nicely. So you can see follicle at day 0 surrounded by granulosa cells, and it expands, forms sort of antrum. The problem is that you can see that the structure of the follicle flattens as you culture them on flat surface. So, and then we kind of depict schematically what's happening. So the granulosa cells that surround the oocyte, and it's very important that the granulosa cells are connected to the oocyte. They migrate away and attach to the bottom. So this approach is successful in mice. However, when you try to culture anything above mice, so cats, dogs, cows, sheep, goats, non-human primates, or humans, this approach doesn't really work. So you cannot get a fertilizable oocyte when you culture follicles in 2D. So a solution was proposed by Shea and Woodruff collaboration back at Northwestern University about 15 years ago. What if we take an ovarian follicle and we encapsulate it in a hydrogel? So whenever I talk about hydrogel, you can either envision jello from the supermarket or some sort of soft matrix. So what if we, and soft matrix is here the gray circle around the follicle. So what if we encapsulate the follicle inside the matrix and then the follicle can grow? Obviously, as an engineering design, you have to think about a lot of things. So you have to match the physical properties. The gel has to be soft enough to allow follicle expansion, but it also has to be rigid enough to support this 3D structure. You have to think about pore size, right? So everything's coming in and out by diffusion. So the oxygen, nutrients, hormones have to be able to go in and out by diffusion. So the pore size is important. Gelation mechanism is important, so it should be not toxic and physiologically relevant. So we isolate follicles from mice. We work with mice. So we use prepubertal mice, 12 to 14 days old, isolate mechanically follicles, encapsulate them in culture. The first hydrogel used was Alginate. There have been, 100, 200 papers on culturing follicles in Alginate. It's a wonderful system. Alginate coming from plants, so it's a plant-derived polymer. It's non-degradable by animal cells, obviously, but you can make it soft enough. So we're making Alginate at 0.25% concentration. So it's a very soft hydrogel, and it can support follicle culture. How do you control the stiffness of Alginate? So here you see the expansions of follicle diameter as a function of time. And if you encapsulate follicles in a very soft hydrogel, so somewhere between 0.25 to 0.5, then follicles grow very nicely and they reach relatively large size, and then they can be ovulated and they can actually develop into live birth if implanted in animals. But then as you increase the concentration of alginate to 1 or 1.5%, then you start seeing decrease in follicle expansion at follicle size, and then they really don't grow. So what's happening? And what's happening is kind of drawn here. So in the beginning, you have follicle in alginate, and then follicle starts to grow and expands. But then as it expands, because alginate is non-degradable, it senses compressive force. And if you have a soft hydrogel, as here at low concentration, it allows expansion. But as you increase the concentration, it doesn't allow follicles to grow. So it works for mice. As I mentioned, you can culture follicles, get them to antral stage, then remove the oocytes, fertilize the oocytes, implant it in pseudo-pregnant mom, and get live births. So why can't we do it with humans or monkeys? And then the explanation is here in this slide. So as I mentioned to you, alginate is non-degradable. So as the follicles grow and expand, the follicle inside would sense compressive force. So you can find this golden ratio for mice follicles. And mice follicles start at about 120 micron in diameter, and then they grow only to 350. But in human follicles and non-human primates or bovine follicles, they start approximately at the same size, but then they reach somewhere between 1 millimeter to 20 millimeter in diameter. And no non-degradable gel can really accommodate this kind of expansion. So what can you do? So one idea that we had is, what if we design a degradable gel? So let's say we're going to put a follicle, and then as follicle grows and secretes proteases, so enzymes that degrade certain sequences, the matrix around the follicle is going to break down and follicles can grow. So we used a hydrogel that's used in a lot of tissue engineering approaches. It's called polyethylene glycol, PEG, and then this is the structure. So it has reactive arms on the PEG, and PEG is really non-reactive, sort of blank slide hydrogel. And then we cross-link it with cross-linkers that are peptides, and these peptides have protease-sensitive sequences. So this is basically a sequence of amino acids. It's about 15 to 16 amino acids. And in the middle, you have a degradable sequence. So if I encapsulate follicle in this kind of hydrogel, follicles inside can secrete matrix metalloproteinases in this case, and then break down the gel. So we've tried that. And then we used plasmin-sensitive sequence, because we know that there is a lot of activity before ovulation by secreting plasminogen activators. So we used this sequence, and then we encapsulated follicles, and follicles actually grew too much. They chew up the whole matrix and fell on the bottom of the dish. So we played a little bit more with different sequences. We also use MMP9 sequences, so matrix without proteinase 9, or we can use MMP2 sequences, so you can really play and find the sequence that would work for your species. For mice, we went back to the plasmid sensitive sequence. So this is a native sequence found in fibrin gels, like blood clot. And then this YKNR was too fast. So we just changed one amino acid to serine here, and you can see follicles can grow, and then they reach, and they grow from 130 to 350. They can be fertilized, and then they develop into blastocysts as well. If you change another amino acid, then it's kind of too slow, and this is a non-degradable sequence showing that there's really dependence on the degradation sequence that you use. So it's either too fast or too slow. So you can find the best sequence that could work for these specific species. But then going back to the stiffness of the matrix, I've showed you before, so the argument here that follicles can modulate their environment by secreting enzymes that break down the environment. So I've showed you before that alginate, so this is the graph that you showed before, we control the stiffness of the alginate by just controlling the concentration of the biomaterial. And then we know that this grows really nicely, and this is the stiffness of this hydrogel is only 300 pascals, so this is super soft. As you go to 2000 pascals, then the follicles cannot grow because it's not degradable. So what happens in PEG? In PEG, all the hydrogels that we would encapsulate follicles would have somewhere between 2,500 and 3,500 pascals, so it's super stiff compared to here, right? But then the follicles would grow nicely, which kind of proves the point that follicles can modulate the environment and break it down. So this is our results with secondary follicles. Then the question was, okay, secondary follicles is great. Can we go and try to grow primary follicles? A paper published in Tissue Engineering from Shea Lab showed that if you take primary tiny follicles, anywhere between 80 micron diameter to 110 micron diameter, and you co-culture them with either mouse embryonic fibroblasts or stroma cells from the mouse, they really do better. So you would have a follicle floating in alginate, and then you would have mouse embryonic fibroblasts on the bottom. And then some sort of soluble factors that are secreted by those helper support cells do help follicles grow. So you can see that without any cell support, all the follicles die by day 10, of any size. And then if you co-culture them, those that survive do grow nicely and reach antrum stage. But then we said mouse embryonic fibroblasts are great, but can we come up with some maybe better or translational cell line? So years later, we decided to use adipose-derived stem cells, right? Because if you think about the patient, it would be probably ideal to take the follicle from the patient and then some fat and isolate adipose-derived fat stem cells and try to co-culture follicles with these adipose-derived stem cells. So we did very similar work. We tried to encapsulate primary tiny follicles with adipose-derived stem cells, and you can see, so all those dots is cells, and the circle in the middle is follicle. We would put it in alginate, and it would grow nicely with improved antral formation, and then nice survival of the follicles. But then going back to the beginning of my presentation, we know that human follicles don't really grow in alginate. So what can you do? So this is just to show that follicles survive better. So I would go and I would try to do the degradable hydrogel. Can I put, so then the question is, can I put primary follicles with adipose-derived stem cell in the PEG hydrogel that I just designed for secondary follicles and make it grow? And the answer was that I could do this, but it wasn't so great. So when we put follicles, so primary follicles with adipose-derived stem cells, they kind of did okay, but not so great. So the survival was worse. The growth was comparable, but again, not so great. So we couldn't understand what was going on. Why does it work in alginate and it doesn't work in PEG? So we hypothesized that probably the environment is too stiff. So we're using 2,000 to 3,000, and then when secondary follicles could degrade it, it was too stiff for the adipose-derived stem cells because they either cannot proliferate or they sense the environment is too stiff. And you probably know that adipose-derived stem cells can differentiate into different three lineages. They can be bone, cartilage, and adipose. And when you put it in a too stiff environment, they do become bone. And probably the soluble factors secreted by the bone cells are not so great for the folliculogenesis. So we designed a dual degradation mode. So we know that follicles love the plasminogen sensitive sequences. So just YKNS, the plasminogen sequence, wouldn't work for both of them. So we added MMP sensitive sequences. So now we have a hydrogel that has YKNS for the follicles and MMP9 for the adipocytes. And to our happiness and excitement, we saw that so this is live dead stain for the adipose-derived stem cells, showing that the ADCs survive much better in the dual hydrogel compared to the pure plasmin-sensitive sequence. And then when co-cultured in this hydrogel that has the dual degradation, so one for the ADCs and one for the follicles, we do see that the follicles survive much better, and then they also grow nicely and they can be reached to maturation. So this is the first part of my talk, and this is kind of focused on the in vitro studies, and we're still pushing forward to try to develop better degradable sequences that can hopefully also help primordial follicle development. So in my second part, I do want to focus on the transplantation. So what can we do for those population of patients that either cannot benefit from ovarian stimulation, or those that have the risk of getting cancer cells upon transplantation. So the idea here is taking this cortical strips and then isolate all the primordial follicles. So basically clean the primordial follicles from everything else that can be there, and then offer this patient an engineered matrix. So again, we work with mouse model, and then the mouse ovaries are pretty nice place to work because the ovary in the mouse is surrounded by the bursa. So if we're trying to ovariectomize the mice and then have the space open for implantation of the artificial ovary, we usually just scoop out the ovary, we put in inert alginate bead to keep the space open, and then 2 weeks later when the mouse shows menopausal symptoms, we go back and then implant an artificial ovary here in that space that potentially can actually let us test pregnancy. So the first matrix that we used was fibrin, right? So if you think about it, blood clot is translational material. You can just use it. It's used as surgical glue. So we would take ovary, we would clean it out as much as possible from all the potentially contaminating cells, and we would just have primordial and primary follicles, and then we would put it in a matrix. So why do you need to put it in a matrix? We know that if you just inject the cells anywhere in the body, it just gets lost. So you do have to provide some sort of initial material to support folliculogenesis in the beginning. We implanted those artificial ovaries, and you can see so some of the follicles, all the follicles are encapsulated in fibrin, and then so we have some early secondary and a lot of primordial follicles, and then we follow the FSH levels in mice. So once we ovariectomize the mice, the FSH levels go up, right? Because there's no estradiol, there's no negative feedback, so FSH goes up, and then we implant the artificial ovarian construct here, and then the FSH kind of stays high, and then it drops. Once we examined the explanted artificial ovaries, we did see that most of the primordial follicles were gone. There were some secondary follicles, but then it wasn't a real ovarian tissue. So we hypothesized that probably fibrin degrades too fast. And if you think about clot disappears from the blood pretty fast. So which means that fibrin doesn't really mirror the development of follicles, and we needed something else. So obviously, if I need something that I can control the degradation, I would use the polyethylene glycol gels, where I can control the degradation kinetics of it. So we used the same, very similar system that we used for the follicle culture. And here, so this is day 0. We encapsulate primordial and primary follicles in this hydrogel. This is the bursa. We implant it, and then 14 days later, you can start seeing larger follicles developing. This is all gel, and this is the bursa. This is the fat surrounding the bursa. And you can see here the larger image that we start seeing some follicle development. 30 days after implantation, again, this whole tissue construct becomes larger, and you start seeing infiltration from the cell from the other side, and you see large antral follicles, all the way to 60 days. 60 days, you start seeing real regeneration and remodeling of the implant. Again, still we're seeing a lot of antral follicles. We also saw corpus luteum forming. This construct was revascularized. We saw functional blood vessels with red blood cells in them. And then looking at the FSH levels, very similar. So again, FSH goes up as you overreactomize. And then if you don't implant anything, the FSH stays high. But then after implantation, you start seeing a decrease in FSH that stayed low up to 90 days of this study. And then we saw so in the beginning, you have the majority of the follicles are primordial follicles, and then about 30% primary follicles. As you continue your experiment, you start seeing development of antral follicles and kind of maintaining the normal ratio between the smaller follicles and the larger follicles. So the conclusion from this experiment was that we can really have a matrix that can mimic the regeneration and remodeling of the ovarian tissue. And then the last part of my talk would focus on the leukemia patients, but I'm not talking here about the fertility preservation, but more about the ovarian function, endocrine function preservation. So if you think about 40 years ago, the majority of leukemia patients didn't survive that well. Now 85% do survive, and because they're pediatric patients, they have a way longer life to live. And but unfortunately, because of the treatments before puberty, their ovaries are not functional when they have to undergo puberty. I like to show this image. So if you look at these two women, do you think they're related? So these two women are identical twins. And then the woman on the right was diagnosed with leukemia when she was 3, and she obviously undergone treatments, and she's infertile, and she has a whole long list of different disease, partially associated with the fact that she doesn't have a functioning ovaries. So our answer to this would be, what if I take an allogeneic tissue, if I take a donor tissue from somebody else, and I will encapsulate this tissue in immunoisolating capsule, very similar to what is done with islets. So in this case, I would have this tissue protected from the host. It's going to be porous enough so the tissue inside the capsule can get oxygen and nutrients and can remove the waste, but it also can receive FSH, follicular stimulating hormone, and respond with estradiol and progesterone. But then this construct is going to play a part in hypothalamus pituitary gonad axis, right? Because it's going to respond to the FSH, it's going to secrete estradiol, and you will have a really normal physiological active puberty compared to passive puberty induced by synthetic estradiol. So again, being an engineer, you have to think about how do you design this kind of capsule. So the capsule has to allow expansion. So follicles are very different from islets. Islets are there and they don't grow. Follicles do expand. I just showed you millions fold in their volume. It should support follicle growth, allow diffusion of nutrients and hormones, and also protect from the host and prevent rejection. So how do you engineer something like this? So I just showed you before that we can design a PEG hydrogel, a synthetic hydrogel that can support follicle development. The problem with this is that because it's degradable, it's not going to prevent infiltration of the host cells, right? So we saw vascularization, which means if it's going to get vascularized, it's going to get rejected. However, so can we engineer PEG hydrogel that's not going to be degradable? Yes, we can engineer hydrogel that's not going to be degradable, but the problem with non-degradable gel is it's not going to allow expansion of the follicles, right? Because follicles grow. So, and we tried, we see here ovarian tissue encapsulated in non-degradable PEG hydrogel, implanted subcutaneously in mice, and then all the tissue was necrotic because it doesn't allow expansion. So you probably already know the answer. So the idea was, what if we make a dual capsule? We would have the core made of degradable hydrogel. It would allow expansion and degrade over time, but then the surrounding shell is going to be made from non-degradable PEG. It's not going to allow infiltration of the cells. And this is how it looks. So we encapsulate pieces of ovarian tissue and in the degradable core, and you can see the border between them. And then we add another layer of non-degradable PEG hydrogel. This is how it looks under the skin of the mouse. This is how it looks at retrieval 30 days post-implantation. So you see the ovarian tissue is white, which means it's not vascularized, and it's easily taking out, it's not surrounded by the cells. So we wanted to test it in an allogeneic mouse model. We took Balb ovarian tissue, encapsulated in this device, and then implanted in C57 black mice. We looked at cyclicity, which is the easiest thing to do. And then you see that without the capsule, if you just put allogeneic tissue, all the mice cycle, and then 3 weeks post-implantation, the cyclicity stops. And if you look at histology, the tissue is rejected. But then if you encapsulate the tissue in the capsule, you see a slow delay in 50% of the mice, but then 3 weeks post-implantation, 85% of the mice were cycling, and then they were cycling all the way to 14 weeks of the experiment. Histological analysis showed nice follicle development. So it's huge, that's why it's kind of here and inside. But if you look at this, so this is the host tissue, this is the whole gel. We have several tissues, pieces of tissue encapsulated, and then at the zoom in, you start seeing antral follicles. This is the interface between PEG and the host, so there is no infiltration from the host. You see some sort of relatively thin capsule formation around the PEG hydrogel, but there is no serious inflammation. And then the big question was, does it induce immune rejection? So even if you see that the follicles are functional, it can still cause immune reaction. So this is the tissue without the protection, so it's completely destroyed. And then if you look at the presence of the antibodies, so we do some flow at day 0, so before transplantation, there is no antibodies against the Balb C tissue, but then after 3 weeks, you start seeing there's a lot of antibodies. This is our negative control, just pure hydrogel. But then if you look at the results from allogeneic tissue implanted in the capsule, you see that, so before the implantation, there is no antibodies, and as you go all the way to 60 days post-implantation, there is really no reaction. So not only that the T cells don't infiltrate inside the capsule, probably the antigens from the tissue don't go outside to induce any immune reaction. So I also had this question that Dr. Latham asked me to ask. So how do you see the use of these model technologies increasing in coming years? And we will talk about this also, and what are the frontiers that we can discuss at the discussion level. So just to summarize, I wanted to show you that the 3D environment and co-culture follicles support the cells, and then that you can control the degradation and really kind of direct it towards the tissue that you're interested in. Synthetic hydrogels can be designed with tunable mechanical and biological properties. So mechanical, you can control the stiffness, you can control the degradation kinetics, and biological, you can really decide what kind of cells you want to use and direct the environment for those cells. Successful folliculogenesis and graft survival, I've demonstrated to you using PEG hydrogels, and then the PEG hydrogel can maintain the protective shell and also support folliculogenesis in the core, and then we also saw low degree of inflammatory reaction. So this is my last slide. Of course, I want to thank my lab. I have a wonderful lab, sunny day in Ann Arbor. I have multiple collaborators, Dr. Lani Shea, Dr. Teresa Woodruff, Mariela Cascaljo is my immunologist, and then this work was supported by the R01 and the NSF Korea. Thank you. Questions? Yes.
Really nice presentation, Ariella. Just a quick question. I'm still trying to get it around my mind. If you have no vascularization when you've got this dual layer, how do the steroids get out? And what happens eventually if that forms a corpus luteum, which I assume you want to do eventually? How are you going to then maintain the progesterone and things like that? That's, without vascularization, how does this all happen?
It's an excellent question. So, so the first part is, how does it happen without vascularization? It's all by diffusion. So the pores are large enough to allow FSH to go in and sex steroid to go out. And we do have, so that's why I have a limitation of the size. So if I would have to do it in larger animal model, I probably would have to put multiple constructs and not one huge one, because it's all diffusion limited. So everything is by diffusion. Your second question was about corpus luteum. So we do see formation of corpus luteum. It does secrete progesterone, and then I guess it just regresses. So, and then obviously there's going to be a limitation of how long it's going to function. So if I'm going to put 100 follicles, it's going to function X number of cycles. If I'm going to put 100,000, it's going to function longer.
Very nice presentation. For your 3D method, so after antral formation, so did you do embryo transfer? Did you get offsprings? Are these offsprings healthy?
This is an excellent question, and this is something that we have to do. It's expensive and long term, but definitely that's why I work with Dr. Hasanta. Yeah, so we have to do it. We do get to blastocyst rate, but I don't have a huge colony to have pseudopregnant mice and to do it. But yes, this is definitely something that we're going to look at. Also epigenetic changes, but also whether the offspring is fertile and so on.
Yeah, I'm sure you know Dr. Epic's pioneering study on this. So compared to his 2D two-step method, so what's your successful rate compared to his?
I think our efficiency is greater. He has about 3 to 5% efficiency all the way back.
I think that's his first study. So he modified his method. He got a quite high rate.
Okay, so I guess I have to look at this. But so is it in humans or in mice?
In mice.
In mice. Okay. So, but if you think about the last paper that came out from Woodruff lab where she tried to culture human follicles in 2D. She had 2 cohorts, so she would encapsulate. So we know that human follicles don't grow in 2D, EPIC method or any other method. But then if it was 3D, you do have to have at least a 3D environment in the beginning. So yeah, I don't do any 2D to even compare to EPIC's method.
Thanks. Oh, can I ask one more question?
Sure.
So what's the strategy for starting from primordial follicles? Because we know in reality, most of the pieces you've got, they count mostly just primordial follicles rather than primary or secondary follicles.
So the strategy, and I will not spill all my guts here, but the strategy would be really designing the matrix. So we can add helper cells, we can add multiple growth factors and degradation kinetics.
Thank you. I'm from Joanne's lab, so we did a lot of the in vitro activation of this. We have kind of in vitro system, doing the activation. I think now to talk to you about collaboration.
Thank you. John Fortune?
Yeah. So I think just to clarify, I don't think your goal is to outdo EPIC. I think your goal is to develop a system where you can do larger mammals.
Yes, absolutely.
In a 3D system, because those won't work in a 2D system.
Yes.
I have a question though. Your schematic, you indicated, and I think just now in your answers, you indicated you would put multiple follicles in a single 2-layer construct. When you do that, and these animals cycle, what is the synchronization? How rapidly do you recruit follicles within that, for maturation? And I guess wondering if you've actually got a nice system for maybe looking at that level of control over normal ovarian functions, controlling the rate of recruitment from the population.
So you're asking me about in vitro or in vivo?
With your in vivo implants.
So with in vivo it works nicely. So whenever I have larger, if I would put only primordial follicles, I'm not sure how it's going to work, but because we implant primary and primordial, I think primary they're already activated and they start secreting AMH, assuming, and then they prevent the activation. So we do see this nice stepwise activation, and we don't burn through all the follicles that are implanted. In vitro, we don't. So in vitro, everything either gets activated or everything dies. So yeah, in vitro we're not there yet, but in vivo it works nicely as long as you have larger follicles that can prevent activation of the smaller ones.
Thank you.